seeing?
You're seeing my screen, and now you're going to see my presentation. And
Okay, now I
go.
feel better.
You can start.
Okay.
Can you
So,
see the presentation, everybody?
yes, now I can see it.
Yeah.
So I would like to announce that somebody told me that it was my job to introduce Jorge, which is a great privilege because he was one of the most catalytic and important people to have ever gone through my lab. And I think he might have been my third graduate student. Is that true? I think it might have been. Or you might have been third and fourth. Jim Wyman and Jorge started at the same time. So anyway, yes, we're recording. So Jorge came to Brandeis in 1986, and that was a tremendous year for science at Brandeis because Jorge came as one of 4 Chileans who joined us for all sorts of political reasons. And the cohort that came included Jorge, who obviously is near and dear to all of our hearts, Alfredo Kirkwood, who some of you know, who's now at Johns Hopkins, John Ewer, who's now back in Valparaiso, and Vivian Budnick, who is obviously still important. So 4 for 4 successes in that cohort of truly wonderful Chilean neuroscientists, and 3 of the 4 are still in the United States. So we're very good at importing talent. When Jorge was always, well, I have to tell you one more thing before I go further, and that is when Jorge came, he had just recently returned from the Antarctica, and he was desperately working on a paper about penguins. And there was this whole drama about this paper because it was in Spanish, and then whether it was going to be in Spanish or English, but it did eventually get published, I believe. In Spanish? In Spanish?
In English. So and there's some famous quotes from the funny English that came in.
Right. I thought, yes. So that was at the beginning of his scientific career. When he started in my lab, or what he catalyzed in my lab, was bringing real state-of-the-art biophysics to the analysis of neuronal dynamics and building models. And I really have all that to credit him with. He, as you know, is remarkably talented as an experimentalist. He has a very strong quantitative sense, and he did a wonderful PhD thesis, which included the 3 Journal of Neurophysiology papers describing the membrane currents in SGD neurons, and then the first really detailed conductance-based model. That was a model which at the time I truly despised, but that's a whole other story. Not his fault. Anyway, then at that same time, he did the remarkable early work defining what we then called the proton current, which became IMI. And that set us off into lots of years of really profitable work. When he finished his PhD,
on that note,
yes, when he finished his PhD, Jorge went off to a postdoc in Paris, and notice that, and I was really, really impressed because he learned French in about 3 days. And then he later went on to several other postdocs before eventually coming back to my lab for a second stage. And it was then that he got to know Farzan and Dirk and the others. So the two epochs that Jorge spent in my lab were 2 of the absolute most golden eras of the work in my lab. And I think a lot of it has to do with his, why would anyone want to do a postdoc in Paris? I think a lot of it was to do with Jorge's experimental elegance, the clarity of his thinking, and his enthusiasm to try all sorts of crazy things, at least most of them, which ended up working. So I really think he's been a remarkable add-on to SDG work and science in general. After leaving my lab, he joined Dirk and Farzan, I don't remember the order of all this, at NGIT and Rutgers, where they now have this remarkable 3-lab consortium where all the intelligence is
introducing this guy who was in her lab.
What do you, somebody should mute somebody.
Yeah.
Okay.
Somebody was muted.
Yes. So, and the rest is history. And so I'm really looking forward to hearing what you have to say. And of course, there's nothing better than IMI. So take it away.
Well, thank you, Eve. That's very generous what you say. I had, of course, the best time in your lab through those years as a student, and then as doing a postdoc after hopping around the world and not finding exactly something else that was better than the SPG world. And so thank you. And I want to start by acknowledging some of my mentors. And since I didn't know that you were going to introduce me, I'll start with you. And I have a picture of you later, but actually, let me jump to that quickly here. Ever colorful, ever smiling, and here on the runway in Chicago, displaying the STG code that's just your students, I believe, made for you. Anyway, I had to put you there, and those are the nice pictures I found. So thank you very much. You have been most generous, and throughout the years as a student and later to this day, including with your presentation here. So thank you. And let me just tell a little story, which is what I wanted to do through parts of this talk, at least. Eve was, when I finished, none of the papers that came out of my thesis were done. And I got this postdoc, thanks to Eve, actually, connecting me to this crazy Argentinian in Paris, and Coco Gershenfeld, that some of you may have maybe heard of or met. And I went there, and she came a year later to Paris to work on the papers and buy some shoes. But we spent, I think, 2 weeks working hard on these papers, thanks to Eve coming actually to find me where to work on. So Paris, I think, was a good attraction, and the papers may be an excuse, but papers came out. So thank you, Eve. During that time in Paris, actually, I met Nino, because one summer I had run out of money, and I found this little European fellowship that took me to Germany to work in Kaiserslautern University, where Nino was at the time. And that's, I think, the first time we met. And all right, so I want to start by actually acknowledging my first mentor, which was Umberto Maturana, shown here. He was my first instructor in college. He taught cell biology, and at the time he was developing the theory of what is a living being. And I'm not going to go into that. He called that theory or hypothesis autopoiesis, meaning self-production. And he always tried to explain everything in terms of the interactions of components. And this day, he's in his 90s, still working. He has left that lab, which he called experimental epistemology, by the way. So everything about reasoning, thinking, and explaining things in very interesting and sometimes quirky ways. Now he thinks about what constitutes society, what constitutes family, and he has become even involved in family therapy, whatever theory of family therapy, and so on. And I put that little quote here that laxly translated says that we have lost faith in the world, he says, but I imagine the humans in general. And as a result, we have lost certainty in things, and that's why we crave control. And as a consequence, we stop reflecting, thinking, deeply thinking. And that's what he does all the time. So he was my first mentor and really influential in my life. And then came Chris Miller when I came to Brandeis, who, this is another little personal snippet here. I worked with Umberto Maturana. At the time, I was training cats and trying to build their visual processes by interfacing the brain and so on. Then I went to Antarctica working on penguin behavior, and I swore I would never touch a single channel. And but the guy in next door, Ramon Latorre, another faculty who worked on calcium-dependent potassium channels, individual channels, contacted me with Brandeis, with Chris in particular, and thanks to Chris and Ramon, I think, and I don't know how much you were involved, I got to Brandeis. So, and then I said, I'm gonna work in his lab once for the first rotation and then forget about channels. Yeah. So the rest of my life has been about talking, trying to understand how individual channels work and produce activities. So that's Chris. And then there, what he said is, okay, so we have this MaxCK potassium channel found in muscle, throw divalence into it and see what happens. And that was the project. And he literally said that, just throw divalence in there. He gave me kind of these bottles of magnesium and manganese and whatnot. And we discovered this effect of magnesium, that if you put it here on the left, on the inside, it makes this channel more and more active. It always requires calcium. If you remove calcium, there's no amount of magnesium that can turn it on. Magnesium on the outside doesn't do a thing. And as a consequence, what you have is this calcium-dependent potassium channel whose sensitivity to calcium greatly improves as you increase intracellular magnesium. So that was my first foray into modulation, in this case, an allosteric form of modulation of ion channels. And then here's Eve, of course. And with that, I want to move into STG world and also mention that a lot of the work I'm going to talk about was done together with Farzan, who has been, to me at least, a great relationship and very fruitful and fun to work with and produce some stuff. So one of the things that we discovered was when one of her PhD students, I think, was rotating in my lab, I believe, and she was trying to clamp PDs, and she found that clamping one PD versus two PDs made a difference, which now I think is trivial. And here we have kind of a basic circuit. If you voltage clamp only one PD, and this is looking at leak currents, you have an effect on the second one, obviously, because they're electrically coupled, right, very strongly. And you measure this current here, the one in black at the bottom. If you voltage clamp both, you measure less current, and that obviously we interpret as being, well, current flows from one cell to another. So if you're clamping from one, you're going to measure a little bit of the leakiness of the other. We're assuming that both PDs are very similar. So if you clamp both at the same voltages, there is not going to be any current flowing through the gap junction, and therefore you're measuring just the leak of the first cell. The same thing happens if you measure voltage-gated currents. With one cell voltage clamp, you always measure more current, and this is shown here in every trace than if you do it with both cells clamped to the same voltages, and on average, here are the IV curves. We built a model to see if this actually made sense, and it does. So the model is of this type of this circuit here on the right, and it does. And the interesting thing that I think was kind of the take-home lesson from all this was this: that if you measure cells currents, any current, voltage dependent or leak or any, in cells that are coupled, you're likely to be making very large errors. And they are voltage dependent because they depend on the amplitude of the current. So here, for example, what we have shown is the difference between these two currents normalized as a function of voltage. And you can see that the error when the current is small can be quite significant. So it's very important to carefully pick the voltages at which you're going to be measuring these currents to minimize these errors, which never drop below about 10% anyway in these cells. So they're significant. As a consequence of looking at these clamped cells, and we were thinking of these cells as being cables with a lump at the top connected to each other. And thanks to serendipity, because Pascal, who did this work, one day showed me data that were completely wrong in scale, and I started putting them in a model, and things came out to be very, very weird. And as a consequence, we discovered this little thing that shows that in electrically coupled cables, you have a very strong dependence in the voltage in the second cable from where you're injecting current as a function of diameter of the cables. A very nonlinear dependence. So here you have two cables, just whatever long, connected by a gap junction. As you would expect, the voltage with distance from the point of injection here drops, as any cable does. A bigger drop appears at the gap junction, and then the voltage continues to drop across to the other cell. But if you look at this drop as a function of the diameter of these dendrites, and in particular the second one, but it works either way. You can change the diameter of either. The second one is the most relevant though. What you see is that, as you would expect, there is less decay, and this is distance from a point of current injection. There is less decay the thicker the cable. No surprise there, right? That's just passive properties of cables. But on the other side of the gap junction here, you have a very steep voltage dependence, and there is a maximum or an optimal diameter where transmission of current has the strongest effect across the gap junction. And it's very easy to understand, actually. The drop across distance here is due simply to passive properties, but the spread across the second channel has to do with how easily the cable allows the signal to propagate. So the bigger the channel, the less the attenuation, and that's why you get this bell curve. It's a conductance matching issue. Very simple, but it has kind of important consequences. And this we explored in more detail with Forzan. And here we have, for example, a cable we played with many configurations. You'll see a couple where we have one cell producing an action potential here, coupled to a cable in the middle, on the end, wherever you want to put it. And the size of this action potential will vary kind of almost in a kind of Gaussian way, kind of a very, very neat way around a maximum or optimal diameter. I mean, not maximum, optimal diameter. So,
okay.
Yes.
Can I ask a question? Paul Katz encouraged me in the chat to ask it out loud. What do we know about the location of the electrical coupling between the cells? You depicted it as remote from the cell body. Mm-hmm. Is that simply because the dendrites, the cells don't touch and only the dendrites touch? Like, what do we know about the location?
Well, at the time we didn't know anything, but I think now from Adrian's work in Eve's lab, we know that these gap junctions happen far away from the soma, certainly not close, not at the soma. At the time, Pierre in France, Pierre Merin had told me that they found also that by disconnecting the tips of dendrites, they could completely uncouple cells. Now, that was never published. Anyway, we had good reasons to believe that the dendrites, or the distal points of the dendrites, are where this coupling happens. Am I wrong, Eve, in saying that Adrian actually did localize them all? I read the paper, but I'm not 100% sure if it was that or glutamate receptors or something.
No, she localized glutamate receptors. The question about where they are is something that dates back to work that John Miller did in Al Silverson's lab, where he tried desperately to find a way of labeling them in the electron microscope. I think we sort of know that they're not up in the cell body, and my guess is that they're at least down in the major neurite or below. But we need molecular reagents.
Yeah, and I believe Mikey has done some fills with neurobiotin, or what was it? A small molecule that actually crosses these gap junctions, and the brightest spots appear to be at a certain distance from the soma. Anyway,
I don't remember that, but I'm glad to hear it.
You don't? I thought so. Making up. But anyway, so yeah, it clearly isn't on the soma, and we don't know exactly how far from the soma it is, but it's early on in the cables, in the dendrites. Okay, and then what are the consequences of this? Here's another little snippet. So for example, if you have a cell that has multiple dendrites of different diameters, an axon invading those dendrites will have a completely different effect on the voltage change that that dendrite will feel or sense, right? So it will attenuate less along the thicker ones, more along the thinner ones, and that's shown here in the bottom as action potentials of different amplitudes and not filtered. But then if you couple those from any one of these dendrites to other dendrites of different diameters, again, you see changes in the shape of the action potential that are not only reflection of attenuation in both cables, but that are maximum at around a optimal diameter, in this case, between this and this, 2 and 5 microns for in this model, right? So it's attenuates on both sides of that diameter. And so thinking that this propagation, the kinetics of this propagation, as you can see in these panels at the bottom, is different and might and seem to be optimal at a certain diameter. We started building little models to look at what influence that may have on the topology of networks and whether they could generate oscillations simply on the basis of gap junction coupling. And so we built, for example, this very symmetrical.
Ron had his hand up. I don't know if he has a question. I apologize.
Thank you. No, please let me know. Yes, Ron.
In the chat. So I want to know how the diameter of the, what is the diameter at the maximum transmission point? How is that related to the diameter of the input cable? Is this a simple matter of impedance matching of the two cables?
Yes, yes, it is. It is. So I forget exactly how it depends on the first cable's diameter, but yes, there are both have an effect on what exactly the diameter is going to end up being. I think, yeah, I can't remember exactly. I guess the thicker the first cable, the more it moves, the optimal diameter moves to larger diameters, I believe, but I'm not sure. Anyway, but the consequences are interesting if you look at networks. And we built, for example, this symmetrical network where each cell has an axon that produces action potentials, a passive soma and 2 dendrites, all passive, and gap junction coupling between 2 dendrites of adjacent cells at kind of the beginning of the dendrite. But it really didn't matter much where, we just chose the beginning. And if you activate an action potential in cell A, what you have is, you have propagation of the action potential. If the current is strong enough to activate an action potential in the next cell, you have a wave of propagation of action potentials from that symmetrically on both sides. They collide on this side and nothing happens. The whole thing just crashes. But if you introduce any asymmetry into these networks, and here on the left, we just put a inhibitory synapse between the first and whatever, the left cell here. What we have is the propagation going in this way, but it can't go that way because of this inhibition. And eventually you have what we call a reentrant cycle, and the action potential can continue to propagate in that cycle, in that ring. And as a consequence, you have kind of a rhythm appear simply because of these things going around in a circle. And that depends strongly on diameter, as these 2 panels show. So for 4 microns, you have a frequency of 27.5 hertz. For a diameter almost twice as big, 7 microns, you have a much slower frequency. And that happens to V cells, and we didn't show the action potential, but they're all like this. I mean, the axon and dendrites, they're coupled just like in the previous one, but no chemical synapses. And instead, we have this cell G kind of loaded with a lot of other cells, so that the current flowing from here kind of splits into these 3 branches, and neither one is able to produce another action potential. So in this direction, the rhythm, I mean, the wave crashes. But on this, in this direction, this one is enough to produce an actual potential, and these two, they converge here, and the cycle begins again, can continue. And here we also have frequency dependence, and that's best illustrated here in this panel, where for the ring with the synapse in black here, we have a very broad range of diameters over which these frequencies, these oscillations can happen. Not so much for the gap junction, purely gap junction coupled quark. Again, all very strongly dependent on diameter. And if you make the network even more complicated, other interesting things happen. So here we build an 80-cell network where cells were randomly connected. We added one dendrite so that we can add a little more variability to how these cells connect. So I think we had every cell connecting to up to 3 cells by random kind of generation of which to connect. Some are not connected at all, and so on. And then one cell is activated, and I think that's cell 1 here. An actual potential is generated there, and this thing starts now propagating in the network and taking different routes depending on how they're connected, which is mapped here. And here is all the 80 cells ordered in order of activation of their actual potential, and you can see that they produce this oscillation. We average the voltage of all of them, and this would be your local field potential, if you like. So you have the whole network, I mean, the network as a whole producing an oscillation in a range of frequencies that is, this is a few hertz. I didn't write it down here, but it's like 5 hertz or so, 4 or 5 hertz. And this is with this diameter. If you change the diameter, this thing always does this, there, in this case, a shorter diameter, everything changes. The order with which the cells activate completely changes, and that's highlighted in red for cells that become part of this reentrant circuit or network. In blue, one falls out of the picture, and the other ones are still there. And the frequency of the oscillations also changes compared to that. So just on the basis of diameter changes, what network emerges can vary, and what frequencies they generate can vary. So both the topology and the oscillation frequency is highly dependent on these, on diameter. So I think that's what I wanted to say about that. Before I talk about IMI, our favorite topic in my lab and some others. If anybody has any questions about this, we can entertain them now, or we go on. All right, we go on. So so that's something that we did several years ago with Farzan a lot, and later, well, you know that. Let me start over here. The neuromodulators obviously affect rhythm generation in the SDG. This has been known for a long time. Most of you, if not everybody here, knows that if you remove the modulatory inputs that come from adjacent central ganglia that contain this host of different peptides in amines, the rhythm tends to either completely stop or greatly slow down. So we know that decentralizing removes a lot of these inputs, and we have always suspected that at least some of these peptide inputs are responsible for this, or peptidergic input is responsible for this. And one of the things that I did for my thesis, as Eve mentioned, was to characterize the current activated by one of these peptides. And later on, Andy Swenson in Eve's lab discovered that many peptides activate the very, very same current. And now we call that modulatory input inward current, IMI, and this is how it looks. And it looks very much like an inverted bell-shaped curve, where we know that the reversal potential is somewhere here on the positive side, because the current is mostly carried by sodium, we think. perhaps by calcium as well, as I'll mention later, but it greatly turns off at negative voltages. Chunbing Zhao, who was a PhD student in Forzan's lab at the time, found out that when you decentralize, in fact, the conductance of, in this case, I think it was LP cells or PD, I forget, I didn't write it down, increases, which is was kind of surprising, although we had kind of an idea what that may be due to, and I'll tell you in a second, but it was surprising because you're blocking a current here. How could the input conductance of these cells increase? But if you look at the currents generated, and here we're looking at a range of voltages between -80 and -55 only, and you subtract the decentralized currents from the control current, so the effect of decentralization at the current level, what you obtain is an IV curve with a negative slope, which we interpreted as being perhaps a reflection of this, right? So that's exactly what you would expect. So the question then was, if, and this is a little equation that describes IMI, whether we could actually recover all the effects of IMI with a single linear current instead of this fancy voltage-bending current. And so we assumed, or we broke down IMI into two components, one that we call negative leak and one that we call positive leak, for lack of a better word, which are simply linear currents that depend on voltage linearly, that match this negative slope portion of the IMI or the positive slope portion of IMI. And we asked, is that a functional, I mean, is that sufficient to recover the function? Is any of this sufficient to recover the function of the Proctoring activated current as we knew it, as we called it at the time, and later was rebaptized as IMI? And so we use dynamic clamp for that. These are the equations that we used for this. And there. And so we took PD in TTX, added the full IMI here shown in red on the left to the cell, and we obtained some oscillations. But we obtained exactly the same oscillations if we only used the negatively sloped portion of IMI. No need of this other one. In fact, if you compare, if you look at these and compare them to the Proctoring effect itself, Proctoring in a decentralized press turns on the rhythm like that. IMI does the same. INL, the negative leak, does the same, but the positively sloped component actually works against it. In fact, increases the leak of the cell. So the way we interpret all this is that the negative slope of IMI reduces the total conductance of the cell and destabilizes the equilibrium, the voltage, the resting potential of the cell at around its equilibrium potential on the left. That is this portion here. Meaning that since this is an inward current, this portion here at the bottom is an inward current, it will push the voltage to the right. But this portion here is an outward current that will push the voltage to the left. But this portion here is enough to sustain oscillations. If we put that in a model where, and you can get the full recovery of the rhythm, the frequency depends on this, the conductance of that linear slope very strongly. And if you put that current in a model with two other currents alone, potassium and H, you can actually produce an oscillator. And the oscillator, the pacemaker of this oscillator is a linear current, which was kind of a surprise, kind of unconventional to say the least. But you can see here that it does work. So a linear current can act as a pacemaker current. It just has to have a negative slope. But that negative slope, as I said before, is risky because on the to the left of this parent equilibrium potential, although the current doesn't really reverse there, right? But it behaves as if it did. The voltage would tend to go to minus infinity. Really, nothing will stop it from going to more hyperpolarized voltages. And the portion to the right of that apparent equilibrium potential will depolarize the cell as you would like for a pacemaker cell. But you need something to stop it from going too far to the left, and that's what IH does in our model. So IH pushes it back and stabilizes the voltage on the kind of physiological range in this case. And on the other side, you have potassium currents that prevent the voltage from going all the way too far. I mean, the equilibrium potential is actually more positive than this. Anyway, so it stabilizes the voltage on the right, on the depolarized end. So yeah, so that was the exercise with trying to interpret what actually about the IMI current is producing the changes in voltage and generation of oscillations that we see in our system. And this is true for any pacemaker current, right? The persistent sodium current, which is known to be, at least thought to be part of the oscillator in many other systems, like in the respiratory networks, for example, have exactly the same shape. So the negative conductance portion of that current is really all that matters. The other thing that matters is it's as I kind of alluded here, is its coupling or matching to potassium currents. In the STG, pyloric network at least, there's only one cell where the negative leak or IMI can produce oscillations, and that is the PD. We never tried the AD, or if we did, we never did it enough, but it's part of the pacemaker group. So only PD, injecting negative leak or IMI into PD produces oscillations, but none of the other follower cells in the network. And our thought was, well, we were considering all these ideas about correlations of conductances that have been floating around in the field, and thought maybe IMI needs to be matched to potassium currents, who in this case, in the case of the follower cells, may be too strong to allow for, or they push the voltage too strongly to the left to hyperpolarizing voltages and stabilizes the voltages there. And INL or IMI cannot depolarize the cell strong enough. And so this is simply to show that injecting IMI or the negative leak portion of IMI with dynamic clamp only generates oscillations in one of the pacemaker group cells and none of the followers. And however, if you block some of the potassium current, in this case, this was done in LP, then you reduce enough the potassium current to allow oscillations as the model would predict. So later on, Xuanbing, and I'm not showing you that, tried to see if potassium currents actually correlate with IMI levels. And that it's very hard to do. We have been measuring IMI under conditions that make it hard to do these experiments because we have been blocking most potassium currents with cadmium for all this work that has been done until now. But recently, Anna Schneider and Farzan's lab, and somebody else before, discovered that there is a much bigger component activated by IMI that is actually the one that is blocked by cadmium. So maybe the story isn't completely finished here in that regard. So I'm not going to talk about that. That's going to be published soon. Hopefully you'll read it there. But there seems to be a matching between these two currents that is necessary and actually expressed at least in pacemaker cells of the STG. So let me turn to the signaling transduction portion of how this current is activated. And this is work done by Mike Gray, who is, I see him here. And let me just backtrack a little bit. At the time I was doing my thesis, we observed that this voltage dependence of IMI, which shows as a inverted bell shape curve, IV curve, is greatly dependent on extracellular calcium. If you remove extracellular calcium, it becomes very linear. At the time, what we thought, or at least I thought, I don't know what you were thinking, Eve, but I think we thought that the best interpretation of this was a model similar to the NMDA channel, in which, in this case, calcium blocks the channel from the outside. So when the voltage is very negative, calcium lodges itself in the outer portions of the channel and blocks all the other ions from flowing in, and that accounts for this portion of the IV curve. When you remove calcium, then channels flow freely, and the IV curve becomes linear. Mike thought, this is. Let me explore something else. He had been looking at the literature a lot, and he came up with this hypothesis. There are these things called extracellular calcium sensing receptors. These are G protein coupled metabotropic glutamate related channels. They're of the same family, and they actually sense extracellular calcium. And so he asked, what if what's happening is that via G protein coupling, probably, this extracellular sensing calcium receptor is what confers voltage dependence to the sodium channel, or whatever, IMI channel, mostly a sodium channel. And could we prove that? And so that was his thesis, and he spent, was it 5 years or so, Mike, or 6, working on this. And I'm going to show only a couple of experiments here, and then stop. But to propose a mechanism, and then maybe open up a discussion of what could be done about this. So the first thing that he recognized and tried to
Jorge?
do. Yes.
Sorry to interrupt. Astrid has her hand up. I don't know if you saw it.
No, but Astrid, go ahead. Oh, there you are.
Yeah, this may be a stupid question. Do we know that intracellular calcium cannot block the channel as well?
Well, no, we don't. That is an obvious experiment to do. Fill maybe the cells with butter or something like that, and see if the calcium, the IMI linearizes, but nobody has done that, actually. I never did it.
Yeah, because that would tie it in more closely with, like, homeostatic sensing mechanisms and stuff.
Yeah.
Yeah.
But let me tell you the evidence we have to suggest that this is actually the mechanism. So one thing that is important is that this receptor is very tightly coupled, or operates via calmodulin dependent mechanisms. So Mike decided to target that first. Well, he also used a activator of a known mammalian, I think, activator of the receptor. That didn't work. I'm not going to show you data, but there is another inhibitor, in this case, blocker, called NPS 123, that actually does reduce the voltage dependence. I'm not going to show you all the data, but let me just show you this portion here that involves calmodulin at least. And so he tried N7. Oh, I'm sorry. So this is what he did. He measured the current with ramps, and the net current is the subtraction of the currents obtained in the presence of proctorin minus those obtained in control before proctorin is added. You get this current here plotted against voltage, and you get something like the black trace here on the right. And he measured the amplitude as an indication of activation of the current at minus 15 millivolts, I think it was, and the slope between minus 80 and minus 20. So he took these 2 points and draw a line. That was the slope, the simplified version or way of measuring the voltage dependence of this current, knowing that when you remove calcium, the thing kind of becomes, the slope becomes positive and linear all through the entire range. And so first he tried W7, a known blocker of calmodulin's effects. And what he found here shown in the left is that at low concentrations, you get some linearization here in red, and at higher concentrations, even further, increase of the slope, becoming more positive. And then over many experiments and in concentration values, he found a very clear and significant effect of W7 on this slope, on this slope of the current, on the this negative conductance portion of the AD curve, suggesting that calmodulin indeed is involved. He tried that with calmidazolium, which is another blocker, so the same effect. I'm not going to show it. And then did this experiment in which he added this molecule here, dinosaur, interesting name, before adding W7. And let me go back to this little cartoon. The way calmodulin operates is by coming off of the receptor intracellularly and being endocytosed. And that releases the effects of the receptor. So if you block calmodulin, you don't allow the receptor to do its thing, basically, namely to activate the G protein. And if you can block this endocytosis, you should be able to prevent the effects of the blockers of calmodulin. And that's exactly what he did. Dinosaur does exactly this. Dinosaur blocks, I don't know, how is it? Forget how it's spelled, but I think that's it. Blocks the effects, and I'll show you here, of W7. So in W7, the curve is linearized strongly. With when you apply W7 at the same concentrations in the presence of dinosaur, there is no effect. And this is summarized in this panel here. All three of these experiments very strongly suggested that calmodulin is indeed involved, and also suggest that what you're saying, Astrid, may be part of the mechanism too, because calmodulin obviously binds intracellular calcium. So it could be through this receptor, and we have some evidence for that because the blocker of the receptor does decrease this the slope. Increase the slope, make it more linear. So maybe a combination of both. We don't know at this point. So this portion then of the mechanism seems to hold. NPS, I didn't put it in here, 123 inhibits the receptor and therefore changes the voltage dependence of the cell, linearizes it as expected. And he did one more experiment here, which is to test with gallene the role of the G protein. So the G protein associated, sorry, this sometimes acts up here for some reason. The G protein associated with the calcium sensitive receptor is a GQ type protein. And gallene is an inhibitor of the beta gamma subunits associated with the GQ alpha subunit. And what he found is that gallene actually also does something, maybe shifts the curve, but over that range on average clearly increases this increases the slope, makes the slope more positive. Again, consistent with the idea that the calcium sensing receptor of the G protein coupled type, like an M-glutamate, metabotropic glutamate receptor type, working via calmodulin and the G protein, in particular the alpha subunit perhaps of the protein, we don't know that for sure, affects the voltage dependence of the channel, which, and I'm not going to show any of that. That was another paper where Mike and this kid, Danny Dodlan in the lab, a very gifted undergrad who's now at Hopkins, showed that it seems like the proctolin receptor in particular, in this case, which is what he used, is apparently also coupled to a G protein, and that the alpha subunit is probably the one that activates some channel, the one that we call MI. But then there is a ton of other stuff that happens. And what we suggest or suspect, and Farzan and Shumbing had some evidence to support this, is that calcium, and Mikey too, calcium apparently also permeates through the channel. And some of that calcium either affects calmodulin, and this is modeling that we did with Yili Zhang and so on, that suggested also that probably a pump regulated by calcium. I mean, not a channel regulated by calcium, like ryanodine channels, and Mike had some evidence for that using dantrolene, influenced both the voltage dependence as well as the activation of the channel. So we have calmodulin here because to avoid all the lines and stuff throughout here, but it could be, and the evidence suggests that CAMK2 as well as this myosin light chain kinase or something similar to that are probably affecting both the voltage dependence as well as activation of the channel. So let me clean this up so that we can actually see it. So this is kind of the model that we entertain now for how IMI is activated and and the voltage dependence of it regulated, involving two G protein coupled receptors. It's kind of crazy, complicated, but the data seem to be consistent with that. And I'm going to say one more word and then I'll stop. But Astrid, you have a question.
Yeah, this involvement of all these intracellular pathways, what does that mean for the temporal dynamics? Like how quickly all of this can happen?
We know that the activation of the proctolin current is fast, right? Although we really don't know that. What we know is that when you change the voltage in the presence of proctolin, the activation or the current changes are very fast. But we have never
that's a voltage response, not a metabolic response, right?
Exactly. So we don't know really how fast the channel is activated or modified, affected when proctolin binds the receptor. Nobody, I mean, we did puffs and stuff like that, but that's never fast enough to really, I mean, we didn't do that, the kinetics of this. That would probably be best done in a reconstituted system or something like that than in the STG, where there are so many barriers for proctolin or these peptides to flow and so on.
But then related to more of the physiology, and this is me, not knowing enough about this, but where and how quickly do the agonists come from?
The agonists are released by modulatory containing neurons like MPN, like MCN1 perhaps, acting on these cells. I'm not sure, Mike would know better if the proctolin that MCN1 releases actually, well, acts on AB, we know, right? I don't know if it does also on these other cells.
It does.
So that's on the time scale of seconds then?
Yes, it does.
It does, right? Yeah, so I don't know if seconds.
It's fast.
It's fast, but we don't really know. Mm-hmm. So let me propose a crazy idea, and I've discussed this with Dave. I don't know if Dave is here today, it's never become anything, but since we are the STG group here and others, maybe all together we can come up with something. So this is do we have a calcium sensing receptor in the transcriptome of the STG? Well, we have a whole load of metabolic glutamate receptors, several of which could be. Type 1 and 5 are the type in mammals at least that form the calcium sensing receptor, or they're very close, very similar to it. Type 3 maybe also, and we have all of those in the transcriptome. So candidates for a calcium sensing receptor abound. We have plenty. So that could be. What about the channel? What could be the channel? And I keep coming back to this one, the sodium leak channel non-selective, Nelson. Nelson is a weakly voltage dependent channel that looks very much like a sodium channel from its primary structure at least, and secondary structure. It only has 2 of these domains where there are enough positive charges in the S4 domain to make it voltage sensitive. So, but it seems to be enough. It has calcium binding domains. It's permeable to sodium. So a lot of things that kind of at least are similar to IMI, but it only works in conjunction with a bunch of other proteins, accessory proteins intracellularly, like UNC75, UNC80, another one called FAM155 in mammals at least. These UNCs were discovered in C. elegans, but are found everywhere. They're very ubiquitous. So we probably have them in our cells, in STG cells as well. But I don't think Dave has identified those. And it's very voltage dependent. And so the IV curve of the Nelson channel, this is a human Nelson transfected into HEK cells, an expression system, mammalian expression system, together with these guys, the UNCs and the FAM protein. When you do that, you get a channel that looks very much like IMI, whose IV curve looks very much like IMI, and is very calcium dependent, extracellular calcium dependent. So when you remove calcium, it linearizes. When you have calcium, it's what I showed you before. So from simply the electrophysiology of Nelson in mammals, one could propose that, did I show you? No, here. And we have Nelson in the transcriptome as well. So we have, aside from these UNCs and intracellular components, we have the main components that could make an IMI type of current. And so we recently wrote this big EDGE grant that wasn't funded, that a lot of us here were involved in, and that could have been a good venue to try to, for example, express these things and rebuild IMI in maybe HEK cells or something, to see if a Nelson is it. We don't know otherwise what other channel it could be. I don't know anyway, and I'm open to ideas. So I'm going to stop here and just say that kind of the calcium dependence things came back to bite my tail after I started with Chris, what is it, 30-some years, 35 years ago at Brandeis, and done a bunch of other things. Smita, who is organizing this thing, is working on decentralized and whatever plasticity mechanisms. I didn't talk about that. And other things we're doing with Alfredo Kirkwood that he mentioned at the beginning, was one of the Chileans who came. Actually, we came together in the airplane to join the program at Brandeis. With him, we have met together to look at correlations in mammalian cells and the cortex, and I've talked about that in previous STG meetings, so I'm not repeat it. But with that, thank you for listening. Sorry it was a little long, but thank you for the questions so far. And what do you think about this Nelson idea? Eve, go for it.
I love it. It actually makes sense. I mean, the molecules are there. And Nino put a note in the chat saying that Nelson is really important in respiratory rhythms and it's highly peptide modulated.
Yeah. I didn't mention that, but yes, Nelson is very important in all kind of pacemaking activities.
So it sounds like it's the best candidate so far. I mean, all the evidence is consistent with, right? So
it seems to be, yeah.
It actually makes a lot of sense.
Now,
Jorge, during your entire talk, I was thinking about Nelson before you even mentioned it, because it's also highly modulated by the peptides and,
yes,
and totally makes sense, you know?
Yeah.
Mm.
So I talked to Dave a couple of years ago about this, and he didn't know if he didn't think he had the full sequence yet for Nelson, but I think it would be nice to have a full sequence to put it into a plasmid or something to express it, or make RNA with and express it in I don't know, oocytes or what have you, or a cell in the STG that doesn't express this thing. And then
it's probably all over. But the other thing is, you know, we've always failed to get antibodies to channels to work.
Mm-hmm.
But I was talking to Steve Smith about something else, and he reminded me that it's possible that if we combine expansion microscopy, some of the antibodies that don't work because the proteins are there at too low concentrations, et cetera, that we might be able to get some of the antibodies that have never worked to work using expansion microscopy. So that's something else to think about.
Mhm,
That
mhm.
if you had the full sequence, or if there, I don't know if Nina knows, I don't know if Nina knows if there are any antibodies floating around. It might be interesting to look at.
I, I,
Nina,
I will contact my I have friends that work on C. elegans on NLCN all the time, and so maybe they have it. So I will check with them and let you know.
yeah, it'd be great. At least, yeah, we have an expert that on expansion microscopy and at NJIT, and you can certainly
Yeah.
do antibody staining, and that'd be great too.
No, but also, I mean, you know, certainly it'd be worth doing some Westerns, you know,
Mhm.
to see. I mean, it could be there in such low concentrations that it's hard to detect that way, but now that you have a candidate, there are all sorts of really interesting things to do.
Yeah.
Um,
Is it in our funding environment, is it worth really putting a lot of effort into this, you think?
Leaving aside the funding environment, the scientific environment, it's worth it.
I'm with you there, but without funding, the scientific part cannot be done either, right? So yeah, I would love to. I mean, Mike, you want to come back to the lab and do it?
If you pay me. I'm looking to get back to the East Coast someday, so.
I'll weigh in on a few things if you don't
Hey,
mind,
Dave.
Jorge.
Yeah. Hi.
First of all, you can have Mike back. We'll put him on a plane tomorrow. No problem.
So that he stops
Get him outta
talking
here.
about MI.
Second, I just emailed you all the basically the expression of Nelson across all the STG cells. It's in your email. If you want to bring it up for the screen now, it's there. And third, I'm pretty sure we have the full length sequence for Nelson if you want us to
Oh, you have
put
it?
together, stick it in a plasmid, anything like that.
Um,
And we see lots of cool stuff going on with Nelson expression when we play around with modulation a little bit, when we turn on the gastric mill, all that kind of stuff. So I think it's a terrific target. And even if we can't express it, maybe we can knock it down or something.
yeah, that'd be great. So let me, let me try and put it here, but I can't stop here. Can I stop sharing and share my screen at home, I was going to say here. So this is a figure that Dave was talking about. Do you guys see it?
Yeah.
So that's Nelson expressed.
And look at it, it's giant in LP.
It's giant in LP, exactly, and then IC as well.
Look at that. I mean, it's in all the right places.
Any reason why AB would be low other than it's really small?
Yeah, you just screwed up.
Never.
Well, maybe AB doesn't express it and everything happens through PD in this little thing.
It could be.
I may have missed it, but here you're showing us the expression levels. What about correlations with other channels?
Give me a minute.
All right. Well, Dave looks for it. Then, Dave, also, if you're looking for things, what about the UNCs? Do you see them anywhere in your?
I don't think we've ever looked at the UNCs, and UNC is such a weird big family. You know, where do we pull them from in order to look? Like if you have a species that you think would be the right place to start. I know Mitch has a ton of them, for example.
Any fees, I think anyone will do because they're pretty well conserved, I believe. But nematodes or flies, maybe other crustaceans already, like I don't know, Artemia, they have it perhaps. I don't know. I'm gonna stop sharing so that they can share and show us whatever he finds. Sorry, non-STGers, this is maybe a little very specific to this, but I think it's an interesting idea that a channel that is known to be involved in pacemaking and other systems like Nelson seems to be also very important here. And it is also modulated, as Nino said.
Mike has a question. Mike Gray.
Jorge, oh, sorry, which
Go
one?
for it.
Mike Gray, yeah, you.
Oh, okay, cool. I really like that you finally found a paper with Nelson having voltage dependence, because when I was looking through it during my thesis work, it was all linear. And is that caused by, what is the FAM protein you mentioned?
Actually, I don't know. It's a
I've never heard
mammalian.
of
This is a paper that came out last year. First author is Chua. I can send it to you or anybody. It's an interesting paper where they reconstituted the human Nelson in these cells, HEK cells.
Do you think the FAM is making it voltage dependent, or do they not know? Or
I think I can't
that'd
remember.
be really cool if they found a way to switch it over to voltage. I guess it wouldn't be a sodium leak channel anymore then. Just definitionally.
Was that? That's
it technically wouldn't be a sodium leak channel anymore, right?
well, the thing is, I think initially it was discovered to be a linear current, and the voltage dependence was not very well characterized. And it depends on extracellular calcium. No, it reduces in calcium, so on. So people, and clearly, I mean, the deficiencies where this channel is gone, it's lethal. So if you remove Nelson, I forget, I think in the
respiratory rhythm is affected by it. I'm pretty sure that the Ren group showed that when they were showing the practicality of it, I believe.
Yeah.
Very, very good presentation. I loved it, but I'm a little biased. So.
Yeah, okay, Dave, do you have something for us?
Okay.
Yes, Nino.
Jorge, very quickly. I also loved the first part of your talk because, you know, we have a lot in the respiratory network, a lot of gap junctions between excited neurons. And we see exactly those kind of fast oscillations, you know, these 5 Hertz oscillations, etc. And they're highly modulated. But of course, we don't have the same detail like you have, but it was very reminiscent of what we see. And then inhibition comes in, of course, so fascinating.
Yeah. Oh, cool. Yeah.
Nobody knows how they're generated. So that could be really like you said with your circle there.
Yeah. Remember also that the theta rhythm in hippocampus, forget his name now, but has proposed that it's due to gap junction coupling between in the axon. So these very fast spindles may be due to coupling at the axon level in between, what was it, pyramidal cells, I think, in CM-CAM1 or so.
Oh, they're everywhere. They're everywhere.
The association with oscillations is not so obvious. Cool. Yeah, Ron.
So I was wondering, you know, especially when in this network that's kind of randomly connected, you showed us an effective diameter on path, but is there an effective initial conditions where you start the spiking, or is it just the diameter and it's a deterministic method network otherwise?
Farzan, do you remember? I believe my recollection is that yes. Huh?
Yeah, it depends. So the same network, depending on where the kindling happens, can produce different oscillations.
Yes, right. That was my memory. Yeah.
So that means that electrically coupled networks like that are chaotic. So that's interesting. The thalamic reticular nucleus, that's like 100% electrically coupled and associated with driving, you know, gamma oscillations in the cortex. So,
right. And yeah, and there, I think maybe the basis of the oscillations could be gap junctions, but then chemical synapses can modulate the topology and things like that. So, and that can all be modulated. So yeah, the sky is the limit. Okay, Dave, I think you have something.
Okay, well, I don't have anything polished, but here's a quick set of plots. So this is PD, for example. Here's Nelson. Seems to like SKKCA. Seems to like Shaker a little bit. Could take a look at, I don't know, IC.
Where is the calcium dependent potassium?
So in IC, it kind of likes Shaker, which is A type.
Not with BK calcium, huh?
No, not. That's LG. Seems to like a lot of things in LG. Calcium channel.
Mm-hmm.
I don't know. There's some stuff there for sure. I seem to remember. This is all Jose's data from my lab, by the way. It's a little bit unfiltered at this point because I just pulled up a quick R window.
Yeah.
Dave, you
Nice.
say, seems to kind of like and stuff like that, but I'm amazed. I mean, these are pretty clear correlations.
Yeah,
Nice.
this is LPG. Again, this Shaker correlation pops out pretty reasonably. Here's a sodium channel correlation that looks pretty strong. So,
And the ones with the potassium channels, at least the calcium dependent one, are there. I mean, maybe not very strong,
yeah.
but it's clearly there.
This is one
Cool.
where, we keep seeing BK and sodium channels actually seem to like one another as well. That's something
That looks super strong.
yeah.
Yeah.
That shows up in I think several. So here's that in PD.
Oh my.
Yeah.
So yeah, Shaker and sodium channel make a good mix. I think Deb showed that's out in the neurite. So there's some stuff to fool around with, I guess. And yeah, Nelson does seem to be one we have the full length sequence for.
All right. Well, maybe we can talk again. All right. If no more questions, then thank you very much. This has
I
been
actually
fun.
saw some underappreciated aspects of gap junctions. Gap junctions always have the strongest correlations. We can't break them. They're always ridiculously strong. So they're always down here in the corner.
But those are with each other, no?
Those are with each other. They just, I mean, it's like they're basically co-regulated across all these cell types. I like thinking about those a lot. That, those are 3 different cell types, and I can't instantly calculate those correlation coefficients for you, but I bet they'd be reasonably strong.
But to make sure I understand correctly, this is correlations of gap junction expression levels
With one another.
comparing between different cells.
This would be within each point is from a given cell. So the 3 annexins in each cell are always really, really tightly
Okay.
correlated in terms of their expression level. Sorry, it's Jorge's talk. I will stop.
No, please. Oops. All right.
I think that was wonderful.
Well, thank you, Eve.
Thank you.
Great, thank you. Who is the next talk?
And I didn't sleep.
So,
You didn't fall asleep. That's great compliment.
so we are going to divert from ST